Am I audible at the end? Alright. So myself Sandeep, I'll be talking about our first tool, MHC Class 1 peptide binding predictions. And this will be the brief outline of my presentation. I will go through the brief introduction, then we'll go through MSc Class 1 binding prediction, the web version of it. That means we also have the other version that Sinu will discuss in his presentation. And we'll follow up with IEDB recommendation for how to choose your method, what is the threshold, different things and the guidelines. And at the end we'll be having a small exercise. So coming to the first part of the presentation is introduction. T-cell epitope, they do not recognize the epitope directly as in case of B-cell epitope, they recognize the peptides that are processed and presented through MHC molecules. So MHC molecule has different percentages for class 1 and class 2, here we are talking about class 1. So class 1 processing is endogenous, that means the peptide for this MHC class 1 is from inside the cell. It may be wild particle or the cell protein or the effective ribosomal particles. So when we have a peptide inside it, like a protein inside the cell, it is chopped down into smaller peptides by proteasome or other proteolytic enzymes, and these peptides are then transported to endoplasmic reticulum. And from this endoplasmic reticulum, they bind to MSc class 1 molecule where they processed and then passed to the surface and presented. When they are presented to the surface, they can be recognized by the T cell response, so the T cell receptors, and they can generate the immune response. And they are very specialized cell, recognized peptide presented on class 1, then the peptide presented by class 2. So CD8 T cells are like specialized to recognize the peptide that are presented through MSc class 1 molecules. Coming to the MSc class 1 molecule, MSc class 1 molecule is presented in all the nucleated cell, and they are specialized cells that recognize the peptide presented by them. And there are two different chain, alpha and beta. And only alpha chain, which has like 3 domain, alpha, alpha 2 and alpha 3, and this is the peptide binding cleft. So in case of MSc class 1, only one chain is responsible for the peptide binding. And if you see this closely, the cleft is closed. So that means only the smaller peptide can accommodate this particular cleft. And if that's the case, that's why like we have smaller peptides that are binding with class 1. I'm not comparing with class 2 because there are larger peptides there. So in that case, only alpha chain that impacts the binding. Coming to the MHC binding prediction, there are many different variants of the MHC molecule. That means there are different alleles that we have. They are the different variant of the same molecules. And the binding of the peptide and MHC is highly polymorphic, but that means one MHC can bind to several peptides, and on the other hand, the same peptide can bind to different MHC molecules. And the experimental characterization of this particular interaction is very costly. That's why we are using the prediction tools. And as Bjorn mentioned about in his presentation, that when we have a data of peptide and their IC50 value, we can use this data to train our machine learning algorithm, and we can extrapolate this data to unknown sequences. This is what we are doing in machine learning algorithms. So how much data we have in IEDB for different MSc molecules? So we have 172 MSc molecule, and most of these data is coming from human, followed by macaque, chimpanzee, mouse, cat, and other species. So you can download this data from this link. If you want to evaluate your prediction model or you want to develop your own model, so you can use this data set. This data is freely available. Coming to the how many algorithms that we have in MSc class 1, these are the different tools that we have. We have the consensus tool, which is a combination of 3 different methods, and these are the different tools that we have. NetMHC Pen, which is recently published, and there are different like AUC value that is reported in this paper, 4 different tools. So these are the tools based on machine learning or the matrix based that Bjorn has already discussed in his presentation, that how we are doing that thing. And these are reported AUC. The AUC value varies from like 0.5 to 1. As it is closer to 1, that means the perfect prediction. So you will see like in case of MSc class 1, we are having like decent performance, that is 0.96 in the recent tool. So MSc class 1 predictions are very close to the perfection.
Can I just ask a quick question?
Yeah, you.
So with your tools, do you actually have enhancements to the specific tool, or do you actually come up with a new version if you have additional data?
I'll go through the web version, and we are like by default we are having the most recent version, but you can also use the other version as well. By default you will be having the latest version. So coming to the MHC Class 1 Binding Prediction web version. So this is where you can find the tool. You know this home page that we are talking about since yesterday. Either you can go on this URL and you can find the tool from here, or you can directly find the tool with this URL. So once you go to that tool, you will be having this page, and this page is kind of consistent with all our tools. On the top, we'll be having different tabs that describe like home, help, or different example sequences. And on the bottom, we have the tool input sequences, where you can provide your input, and here you can select different methods. This page will be consistent with all the other tools. Now I'll be going through all what this tab means here. The home page is just to provide your input sequences. You can input your data, and in the next you will see help, where you will see a tutorial. If you don't know about the tools, then you can learn from here, and what this tool is doing, how it is doing, how you can provide the data. And there are different things that is explained, like what does it one mean, what does two mean. It is being explained detailed in this help app. And if you want to know what kind of example I should have, then you can go to example tab, and you will be having some example already set up. If you choose these example and then submit, it will automatically populate the home page so that you can like go through the tool how it is working. Then next you will see reference. These are references related to different tools. When they're like references based on the ANN, SMM, or different metrics, you can read in the detail of these publications. The next is download. Sinu will go in the details of it, how you can use once you download the tool, and you can also download the datasets from here. Datasets related to MSc Class 1 binding, or you can download the tool itself. Sinu will cover the thing like how you can download and go through the details of each tool. Then if you have any issue using the tool, you can contact us, or if you need any assistance regarding the interpretation or anything, you can contact us here on the help email, or just go there and tell us. Now coming to the home page again. This is our home page, and this is the first thing that you will provide is the sequence. Either you can copy and paste it, or you can upload a file. And the next, alternatively, you can also select the sequences from NCBI. If you click on this link, it will open a sequence browser that will having a pop-up of taxonomy browser in NCBI, and you can select the sequences of your selected organism, and from that you can copy and paste that, copy from here and paste in that particular home page. Going to the next step, once you provide a sequence, then you need to provide which tool you want to use for the prediction. By default, we are recommending like IEDB recommending the latest one, and you can also use the other version if you click it here, the other older version.
So, when you put in the FASTA sequence, does the tool do predictions for all possible 8 MERS or 9 MERS or whatever length you want to do?
Yeah, we will be going to that thing. You can select which you want to
Okay.
choose 8 MER, 9 MER, 15, up to 15 MER.
Okay.
You can choose that. I'll be showing that in the
Okay, great.
Yeah. So by default, we are showing like IEDB recommended as a default. You can choose another method as well from here. And if you want to use some other version to compare your previous predictions, so you can go and select the older version, and it will make the list bigger than that, and it will be showing the other version like IEDB recommended 2.18 or other consensus or NetMSC 4 or 3 or 2.8. So why we are recommending our prediction method, we are recommending as IEDB recommended method, which is a consensus method. There's a combination of 3 methods, ANN, SMM, and COMLIB, or NetMSC, and depending upon the allele you are selecting. Yeah, so advantage of this tool is it's the best available method. These are the best available method, and you take the consensus from this method, and it provides a consensus percentile rank. By consensus percentile, I mean each method is having a percentile rank, and the consensus take the consensus of 3 methods. So the percentile rank reported in IEDB recommended method is the consensus from 3 different methods. It also provides the binding affinity and the percentile rank of each individual method, that is the consensus is taken from, and recommended method may change depending upon our benchmark studies. Now coming to the allele selection, you can choose your allele, then you need to specify the host first, and then you need to populate your allele. Once you select the host, like by default we are selecting as human here, and then you can choose your allele from the drop down menu. And then next you need to choose the length. Like here you can choose the 9 mer for this particular length and 10 mer for this particular allele. You can alternately provide your own allele sequences, own allele names and their 9 mer or 10 mers. Why we are saying like it is important to select 9 mer, 8 mer for particular allele? This is because we analyze a data set where we find like different alleles have different preference for different sizes. Like this allele has preference for 10 mer, 11 mer and 12 mer, 13 mer more than the 9 mers. This is a binding affinity for 9 mer and this is a binding affinity for larger size, and this is a binding affinity for the 8 mers. So different alleles have different binding preferences for different sizes. But if you analyze the ligand evolution data, most of them are preferring the 9 amino acids. So new methods are incorporated using this kind of information and are being already implemented in the IEDB tools net MSc pen. Coming to the allele selection. So by default you will see only the most occurring allele, and if you click on this icon you will also see the rare alleles. If you see your allele is not there, that means you need to deselect this tick mark here. And the other important point here is the reference allele. The reference alleles are the list of 27 alleles that is able to cover 97% of the population. These are those 27 allele and this is their frequency in the population. So if you want to predict your epitope for larger population coverage, you can choose this set of reference alleles. Once you click on this particular icon here, it will automatically populate the 9 and 10 length of each allele in the reference chart. And alternately you can also provide your sequence, and this should be the format of the sequence. You can see this format from the help page, and make sure you will be able to select only the allele that are already there for that particular tool. And you can also provide the allele sequences. You can also specify the allele sequence itself for some tool like NetMSc pen. And this is how you can select the sequence. The next option here is to sorting it. You can sort your output by percentile rank or position in the protein. And the next optional parameter here is what here? Thanks so much. And you can also specify how your output should look like. Either you want your output in HTML table or your text format. And if you want to receive your result on your email, you can provide your email address here. So coming to the, and then once you provide all these parameters, then you can submit it. And this is how our tool works. It breaks down your sequence into all the possible peptides of your chosen length. Like you have chosen 9 mer or 10 mers, it will break down your sequence into those 8 mer or 9 mers and predict the binding affinity of each of the peptide and the method selected. And then you compare the binding affinity to that of the large set of the randomly selected peptide. That is for percentile rank that we are going to discuss in the upcoming slides. And in case of consensus method, it pick the median percentile rank of those methods used. Here we see like this is our output. In the output page, you will see what was your input. This was your input sequence, and in the lower part you can see the link for download result and the citations. So if you go to the download result, you will be landing to this CSV file that you can open in the Excel sheet. And then on the lower end, you will see the link for the citations. And if you have provided your email address, you will see this kind of email, where you will have all the input parameter and the output files.
I'm sorry, could you explain go back and explain the column on the percent rank? Yeah, no, the next slide. Right there, the percent rank column.
Yeah, I'll come to that.
You're coming to that.
Yeah, I'll come to that. So I'm just showing like this is your input sequence itself. Here you can download, and this is a link for the citation. And this is a file that you will receive in your email that you can open in Excel sheet. And this is how your email look like when you submit your prediction. You will also be having your input sequence and the output result and the parameters. Coming to this part of the things like percentile rank. The first thing that we already mentioned, low means good binders. Percentile rank is the rank of your peptide against a large random set of peptides. How good your peptide is binding against that random set of peptide that we have generated. Lower the rank, it is good binder. That means your peptide is binding stronger than a large set of peptides.
So is this the percent rank, is that in the percent of good binding? Or I still don't understand.
Percentile rank is the rank of your peptide
Uh-huh.
against a set of randomly generated peptides.
Okay.
If your peptide is coming in the top 1%, that means your peptide is binding binding better than 99% of peptide.
It's binding better than 99.7% of the other?
Yes. Again, the random peptide, yes. This is in comparison to the large set of random peptides.
So we have a set of collection of randomly selected peptides.
Mhm.
So for each peptide, we predict the binding affinity
Okay.
in terms of activity, and then compare this predicted binding affinity with that of the large set of random selected peptides, and we assign a percentile rank based on this peptide's affinity compared against the other set of peptides. So based on that, we can rank this peptide worthy.
I guess my question is then, for the top one,
Yes.
Mhm.
do 99.7% bind more poorly?
It worthy than this one.
Okay, that's
If you rank
a
it
little
up-
confusing, it seems.
Yes.
It's sort of inverted, but
Yep.
Yeah.
okay.
So the lower the numerical
That's why
value,
we
the better
highlighted
the
here
binding.
like low percentile means good binders.
Okay, well, I understand now, thanks.
Yeah.
So for each entry, so the set of the randomly generated peptide is same or different?
Yes.
Is it same?
Is same. Any more question?
How large is the set? How many random?
I'm not sure about the exact number. It would be in like 100 or something.
10,000?
10,000.
Alright, so this is the concise result, and if you click on to expand the result, this table will be expanded because you have used the consensus method, so you will see the result from the other method as well. As I earlier mentioned, consensus is a kind of taking the median percentile rank from 3 different methods, and these are 3 methods that has been used for this particular allele. These methods can be different for each allele. Like if you don't have, if these methods are not predicting for any particular allele, in that case it will be using net MSc band.
What is the difference between these two IC51, ANN and SNN?
these are coming from the different methods. We have a method called ANN and we have another method called SMM.
Is there any correlation between them?
I'm not sure about the correlation. These are based on different metrics.
Yes, they are highly correlated.
They are highly correlated, yes. I'm not sure what exact correlation. You would know the number. So these are the three different methods that is being used for this particular allele, and the consensus again take the median for an example for this particular.
I'm sorry, are there definitions for each of those numbers in the health function?
Which number are you talking about? The percentile rank?
For each of the result numbers, so the ones that are highly correlated. They're different metrics.
Yeah.
So my question is, are there definitions?
Not different metrics, sorry, there's different prediction methods. So one is a neural network, the other is a linear matrix. The third one is directly measured value. Yes, all three methods are explained, but they're trying to predict the same thing in this case. It's all IC50 values, predicted IC50.
But there are definitions in the
Yes.
in the health
So I mean,
function.
there's references to the papers.
Yeah, we have already.
All of these are peer-reviewed, published.
Right, but I'm asking, does the software itself have the definitions laid out?
Definition of what?
The definition of the explaining what the metric is.
It's always an IC50 value.
Should be in the help page, yeah.
But it's not different though. Yeah. These are all based essentially, these are trained based on peptide binding data that is IC50 values. And they're trying to predict that back, so the definition would look the same.
So moving to the next, how this consensus mean. So for this particular peptide, against this allele, we have three different percentile rank. And the consensus method rank is different from any of this because we take the median of three different methods. So like for this particular peptide, we have ANN has 0.7 of percentile rank, SMM 0.4, and Comlink has 0.2. The median of each is 0.4. That's why the percentile rank in case of consensus is 0.4.
So the ranks are all the similar numbers.
Yeah.
Yeah.
Why the median?
Because we don't want to go for mean or the mode. In that case, we will be biased to one method if there is large difference. That's why we pick the median.
Okay, so actually in the ANN and the SMM, which are highly correlated and perform much better, the complet is kind of crap. So we have this tiebreaker. And you don't want to have, so it just tells you I tend to do because it's as a single one, you can't move the median. So it says it's on the higher side or on the lower side. That's kind
Just along those same lines, how does that relate to, because it's a different kind of set of numbers, how does that relate to IC50 for the combo lab? How would you think about that in terms of, is there any relationship? No.
So it's roughly correlated, but that's the one that is not an IC50 value. That's it's measuring peptide libraries, synthetic peptide libraries with peptide, one residue constant and all the other ones mutated. So it's kind of like an average affinity of the peptide mix. It's hard to interpret what that is.
So moving next to how to choose the binders. So there are three different approaches that you can use for selecting the binder. Either you can choose all the peptide that has rank less than 1. And this is not the top 1% of your peptide. You can also choose top 1% of the peptide. There are some reports that are picking up top 1% of the peptide in their sequence. But in this case, the top 1 percentile means it is the top 1% in case of large random set. And you can also pick a peptide based upon IC50 value, and the general recommendation is like IC50 less than 15 nanomolar is correspond to high affinity, and less than 500 correspond to like intermediate affinity, and less than 5,000 are going beyond a kind of low affinity. And this has been reported by Alex in his 1994 paper. Like 500 is kind of a good threshold for choosing the binders. Or you can choose binding affinity according to your allele, which has been published in a paper in 2013. It has shown like different alleles have different peptide size. That means this allele is binding more number of peptide than this allele. So the binding affinity of this allele will be different. That's why we have allele-specific affinity cutoff as well. If you want to know what's the cutoff of your particular allele, you can go to help page. On this help page, it will list the binding affinity that we recommend for each of the allele. Is that clear? Yeah.
These binders, whether they correlate with functional also, like stimulating the CD8 T cells.
Weak binder?
Yeah, means good binder, whether they will be good functionally activator also of CD8 cells.
Yes. I think you compare the good binder and the bad binder. Is that
Then
clear?
whether they will be presented to the CD8 T cells and they will activate also in a better manner than those which are not a good binder.
No, the method predicts binding.
Yes.
Binding is a,
Class, the MHC.
right. So that is a condition that is necessary, but not sufficient for T cell recognition. So, and that later on we'll get into how binding or eluded ligand predict immunogenicity and processing and so forth.
So it's perfectly possible that you can have a higher affinity peptide binding peptide that has lower T cell reactivity than it's not.
And what's the relationship of the population allele frequency to the specific affinity cutoff? Because the population frequencies of the allele varies greatly in HLA over many populations.
I'm not sure.
For
about
instance,
that.
this would have no, in American Indian populations who are full heritage, these population frequencies would have no meaning because they're
Yeah, so this
very
it's
different.
it's an attempt to do a worldwide population frequency. So merging all the data that we have available.
And the reason the population frequencies, the reason the population frequencies are merged in with the cutoff data are?
That's kind of random. So we are reporting here those cutoffs. We are focusing on alleles where there's a reasonable worldwide population frequency. There's obviously many more. There's thousands, as you know. And we are ignoring here alleles, and we don't have this data for alleles that are rare because essentially there isn't enough epitope data available for them that we actually do these other estimates. So it's just actually, what was the threshold here? 10%, 1%? I forgot.
So this represents the frequencies in your database that you're comparing with?
So the two columns are not related to each other.
Okay.
They're totally different variables.
All right.
One, the population frequency is just basically telling you worldwide that particular HLA how frequent it is.
On average.
On average.
Okay. But
The other column says for that particular MHC, what is the affinity cutoff to predict binding and for T cell epitopes. So there is no reason why a MHC molecule, which is very frequent, needs to have a high or low affinity threshold. And likewise, an MHC molecule that is not so frequent could also have a high or low affinity threshold. Those two are totally not correlated with each other, but they are shown there in the table next to each other for the sake of convenience.
One question, the cutoff that you are putting there, they correspond with 1% of ranking score, is that right?
Sorry, can you repeat your question?
Okay, so you have a cutoff over there in terms of IC50.
Yeah.
You know, but when you see that peptides, the recommendation is to select 1% of ranking score.
Yes.
Okay, so that cutoff corresponds to 1%.
This is the IC50 value to choose binder against this particular allele, and they are coming in 12%? No? Sorry, you can explain that, yes.
So this is, so that's a general recommendation that we provide 1%. So this is for these particular alleles. If you use this cutoff based on IC50, it's a different.
But when you select IC50 for A0201, 255, you know, I guess that you roughly are, putting a 1% cutoff of ranking score.
It's experimental.
No, it's
It's not predicted.
data from your paper, Senior, right?
Yes.
So this is based on experimentally defined T cell epitopes.
Yeah.
So this is not the 1% equivalent.
No.
That's what he was saying.
Okay. Yes, this is an independent, separate, different cutoff. It's not equivalent to that one.
So
So
we
this
looked
is a
at
better.
we looked at the epitopes in the database, and looked, I think it was 80% or so. We asked at which threshold do we have 80% of the T cell epitopes covered for a given allele, and that's the affinity here, which mostly corresponds to roughly 1%, but not always. And that was essentially some alleles have generally high affinity, so and others less so, and that kind of seems to drive this. So we don't have this number for every allele because many alleles aren't studied frequently enough that we can do this analysis. If you only have one epitope, you can't say what is the 80% threshold. So we're only reporting it for those alleles where we have enough data, and that happens to be the ones that are frequent in the human population. That's why that's in the same table, but I agree it's kind of misleading.
I understand, but can you go back? You know, I'm sorry to miss it. You go back. Okay, back. No, back.
This.
That was the main.
Back. So right. So you have go to the right, please.
Next.
Yeah, next. Okay, so now you have percentile rank 1.7.
Yeah.
Okay, and this is computed from comparing the score of your peptides and the score that you have actually pre-computed for 10,000 peptides.
Yeah.
Okay, so it's something quite easy, But at the end of the day, you could, because you have that computed, you could say, that percentile rank of 2.0, because you are calculating that with regard to a reference data set, will also correspond to one EC50 value. So that's my question. Do you understand what I'm trying to say? So you can correlate that 1% with a particular value as well. So that's why I asked whether, there must be, for sure, a correlation between that 1% value and the EC50 value, sorry. So that's why I asked, whether that kind of corresponded to a number that will give you the 1%.
Yeah, I don't think we have a specific
Maybe
number that can correspond with this 1% rank. Yeah. So for each allele, so in that work, that what we found out was, for each allele can have different repertoire size, and based on that, we can have different levels, cutoff threshold levels. For each allele, it can be different. So we are giving, for the alleles for which we had data and we analyzed, we are giving those cutoff values. So that's independent of this one. So that's a different level of cutoff, different type of cutoff, we'll say.
Okay.
In like we are saying there are two ways to select your binders. Either you are going for percentile rank or going for IC50 value.
Yeah, okay, sorry.
So
and now I have also another question. Can you go, can you go ahead?
yeah.
So it's related to the size, actually.
Yes.
It's quite interesting. So the number of high affinity peptides for every allele,
Yeah.
how is that related to the actual data sets? Okay, so this is my point. A0201 is one molecule that has been studied quite a lot. So the number of peptides that we have for that allele is much greater than for other alleles. So how do you discard that there is a bias,
In the data
bias
set itself?
related to the size of the data set?
Yeah, they can be biased in the data set too. But on the exact data set here.
Well, my point is that A0201 has been studied quite a lot, so you have hundreds, tons of peptides that people have identified that they bind to A0201. So at the end of the day, so you report that allele actually is binding more peptides. So this is what you are reporting. But my question is whether this is only a fact due, or this is only due to the fact that people have investigated more that allele, and you
I
have
think
more
So
peptides.
yeah, the no is the quick
So
answer.
the question is-
So the next best study is A0101, which is here at the very right. So roughly what this corresponds is the rareness of the amino acid motif. Like whereas P5701, for example, needs the tryptophan at the C terminus. Tryptophan is a rare amino acid, so you end up having much fewer peptides that bind well. Versus A0201 is very promiscuous in terms of it can have essentially every hydrophobic residue at the C terminus, so you just have more shots on goal. That ends up roughly what this is.
Has
So
anyone
there
done the-
could be a study bias, but I think in this case, it's
Has
probably not.
anyone done the following study? So you take, let's say, a random protein, 1,000 amino acids, whatever, not random.
Exactly, and then you
chop,
measure
chop
affinity.
the proteins
Yeah.
in 9 amino acids. And
It's
then,
essentially, so when we do the
but people have
combinatorial
done that,
peptide,
you've done that.
so we've done something better, I would say. We are measuring affinities of combinatorial mixtures of peptides, which are essentially billions of peptides at the same time. You can use the same mixture measure against the different alleles, and that correlates perfectly with this.
Okay, so there is experimental with that.
So to further elaborate on that, the point you make that there are more A201 described peptide epitopes because people have studied that is correct. But this is looking at all the different A201 epitopes and asking what is the binding threshold that is necessary to capture, say, 80% of the epitopes. So obviously, you're correct. In the case of A201, if you have 1,000 epitopes, that threshold can perhaps be defined more accurately as compared to an allele for which you have only 3 epitopes, in which basically the threshold is not defined accurately. And that's why we also took note of the frequency of the HLA, because HLA that are not frequent are not studied, but there's not a lot of epitopes. So, but everything is relative of, for those epitopes, what is, for that MHC, what is the affinity threshold that captures 80% of the epitope?
No, I understand, but that figure will be much better because you're actually answering the question that we don't explain. And my one is that frequency, that in the y-axis over there you have a fraction of percentage of predicted binding. And then I see will be fraction of actual predicted binding. And we do that is with the experiment. And again, having a random protein, chop the protein with miners, and then feed those peptides to each one of the molecules and really determine binding, and then say how many of those really each allele is binding. I mean, because at the end these are fraction of predicted. So as long as I see over the fraction of predicted, somehow, but again, I mean, this is already answered. You don't experiment.
Okay, we'll follow more on that question later. Let's go for the quick exercise.
I have a short question. So in terms of selection of binders, you can either choose percentile rank or binding affinity.
Yeah.
Which do you recommend?
Yeah.
General recommendation is percentile rank, less than 1.
So this percentile rank has like a mix of like experimental data. I mean, it's predicted, but
Yeah. It's predicted. So, question is?
So as for me, I would have used like number 2 because maybe there's a comparison with experimental data. So why choose percentile rank?
You can also choose top 1% of the peptide in your case. How many peptide you want to synthesize and go for the next stage. In that case, if you either qualify against this rank.
Can you go to the slide, to the next slide? Okay, this says essentially what percentage of peptides, if you randomly choose them right in based on the prediction, how many would you choose? So if you would do 500 nanomolar at this threshold constantly, you would pick for A0206 10% of your peptides. And for B5101, you pick like 0.1% of your peptides. So you have like 100 more peptides that you would test for one allele than for the other, and totally bias your approach towards that one allele. And what we end up finding is if you do this, even though those all have the similar affinity, the epitopes are nearly exclusively at the bottom there. They still remain in the top 1%, roughly, right? So this is the advantage of the using the percentile rank. You're kind of treating different alleles similarly. And what we find ultimately is that even though B5101 or these other alleles on the very right-hand side, you tend to find more epitopes that have lower affinities. Versus for the one at the very left-hand side, most of the epitopes are actually in the very top. So you end up having, so that's why the nanomolar threshold makes it harder to compare. 1% is better, and it's really easy. And ideally, you would do the analysis that we've done for a few alleles where we have enough data that we did on the last slide to really say what is the experimental threshold that is appropriate for each allele, but we don't have enough data to do that for everyone. That was this next list on, I think, the next slide, where we actually have specific affinity thresholds that work the best. And overall, 500 nanomolar works well, so yeah. So I really need to do a better job with this. It's horrible. We should have a clearer recommendation. But 1% is ultimately what we do ourselves because it's easy, it's unbiased. You represent the different alleles in the same number of peptides that you're testing. Yes.
All right, so going to quick recommendation. Either you can use binding affinity or the percentile rank again, or you can use allele specific threshold. And the next is time for quick exercise. We will not follow up the again thing, like we are not going for the demo thing. So we just show like when you have this access in the protein from this access number, can we predict the all the peptide of 9 length of this allele. So you can find this sequence from the NCBI and then you can submit it to tool and you can choose IEDB method as the default IEDB recommended one, and you can specify the allele from selecting human as a species and finding the allele in drop-down menu and choosing the length and then submit.
Actually, I have another question.
Can we maybe do this first?
Okay.
We can do the questions later because
I
we're
think
way
we are running out of time, so
yeah.
are we going for exercise?
We're already over, so I would say skip exercise and we can do exercise either during one of the
I'm
breaks.
just showing a snapshot of the exercise.
Yeah. Okay, go ahead.
Okay then,
Run through it.
you can submit the sequence here and then you can find the IEDB method by default is selected. Then you can specify the sequence from human and then finding the allele in this drop-down menu and choosing the length. Once you have selected all these parameters, then go and submit and you will see some result like this, where this is showing your input sequence and this is showing your output result, and this is the peptide that is binding with this percentile rank, which is the top binder because already been sorted by percentile rank, and you can, I highlighted this particular peptide in the input sequence, that's there. So you can do it by yourself if you have time. I think we are running out of time.
Is there another one to run through?
There is another exercise too, but it will be high enough time.
If you want to go through it, if you have slides up there,
Okay,
you can run through
yeah,
it.
I'll start it. So in second exercise, which is taken from this particular paper, we shown a malaria antigen, this particular antigen, and in this antigen is shown to induce both response like CD8 and CD4, and our goal is to find the minimal epitope in this 15-mer. And we have HLA typing data there. So the other statement is, can we use IEDB recommended method for this peptide, and we already know the HLA typing of the donor. So you can copy and paste this sequence and choose the IEDB recommended method as default, then specify the species and find the allele there and choose 9 and 10 as the length. And when we do all these parameters set up here, the sequence, all the alleles, IEDB recommended is by default is there, and then submit, then you will see this kind of result. Is it clear?
Sorry, can you go back to the previous, so that's not a genotype then, it's the A, because you have 4 A alleles.
4 alleles here. These are the HLA typing from different donors.
No.
Oh, I see, no, they're
Same
different
length.
lengths.
But some are
They're
nice.
different length, okay.
Okay, so you're doing,
9 and 10.
yeah.
Because the problem
Got it.
statement we said length 9
Yeah.
and 10. So we can provide data like this and you will see it like this also. These are the peptides that are binding to these allele and these are the percentile rank. So by default we are choosing the top 1 percentile. So this is the cutoff and you can choose this 3 peptide because there one peptide is binding to 2 alleles. Is that clear? So with that, thank you very much. And we'll take any more questions later.
Yeah.
Yeah, I have
Thank you.
So
Down.
if you have the same and choose 2 different sites, you duplicate the number of predicted peptides.